Hey everybody. So today we are going to talk a little bit about the neuron structure. So what our neurons actually look like, and when we get to understand the anatomy of the neurons, it helps us to better understand the physiology of how they work. So with that said, let's start with a few terms you may or may not know. First one is that the control center of the neuron is known as the soma. So the cell body that we're most familiar with, I'm going to keep going to and from these pictures. Right? So here's a cell body up top here. Here's the cell itself. Another term for this is the soma. So it is very important to understand that there are two ways of addressing the cell itself. So that is the soma. Inside the soma, we're going to see that the nucleus and nucleolus are present. This is something that's familiar with many cells, that they have the nucleus, and as well as these Nissl bodies that are located in the soma. Those are the rough endoplasmic reticulum that are located inside of that cell. These, and along with all of these components, we're going to have the supportive neurofibrils that are going to be really important for making sure that there is a strong base for the different parts of the neuron, such as the axons. So, really important. These are really important structures. As these neurons mature over time, they're going to lack centrioles, and that's important because if you remember centrioles, these are components of the cell that are needed for mitosis. And because they do not have these centrioles, they are not going to divide. There's going to be no mitosis occurring after their adolescent phase. So, in other words, when they become mature, they stop dividing for the most part. If we look into the cytoplasm of the neurons, we're going to start to see a few non-membrane bound components, and these can be glycogen granules for energy. We can see some lipid droplets, melanin, and lipofuscin, which is a pigment found in the neurons. Dendrites are going to be the cellular extensions that come off of the cell body of the neuron, and we can see that in this image here. If we're looking at the soma, the cell body of this neuron, there are going to be many dendrites that come off it. There may be one, there may be multiple. In this case, there are many dendrites that extend off of the soma. And at the end of these dendrites are going to be receptors. These are really important for communication because other axons are going to release neurotransmitters that will attach to the receptors of these dendrites. So these are only going to be used to receive signal, which is really important. On the opposite side of this, of the soma, we have the axon hillock, which will give rise to the axon. So if you look over here, again, I'm only skipping just because it's a little bit easier to see in this section. The axon hillock is this section right here, and it's going to give rise to the axon, which you can see continuing this direction. Here's the image with that same information. I just thought this was a little cluttered, that's why I didn't show it. So this is really important because at the end of the axon, it's the opposite of the dendrite. They're going to have the synaptic bulb, which is going to have those neurotransmitters housed within them, so that when a action potential travels down the axon, the neurotransmitters can be released from the synaptic end bulbs of the axons. Once those neurotransmitters are released, they can attach to dendrites of another cell. Something else to keep in mind, the large neural fibers. If you have larger neural fibers, they are going to conduct impulses more rapidly than the smaller ones, and therefore they can be considered a little more powerful because of that more rapid conduction of impulses. Not only that, but there is a difference between myelinated and a non-myelinated axons. Those that are myelinated, as we mentioned earlier, will be faster than those that are non-myelinated. Here are some images with some of the terms that we just discussed. And as we look, you can pause this if you'd like to. And if you look over here, here are additional images of different parts of this, the axon in this case labeled. We did talk about this previously. But if you do have any questions, please let me know.